launched 3 platforms over the history of our company. So we're based in Seattle, Washington. And the first system that we launched was a bulk analysis system called the Encounter. So this is a single molecule imaging-based kind of platform. So there's no enzymology. We work in FFPE tissue. This is the first, really a foundational chemistry and platform that laid the way for our Geomix digital spatial profiler, which I'll cover briefly today. You'll see some applications from Sarah, Arutha, and Melissa. We'll talk about some data sets as well. And then also I'll focus primarily on the CosMx Spatial Molecular Imager, which is our single-cell subcellular in situ imaging platform. So this is a very exciting space, obviously, to be in. We've had many speakers here today talking about these technologies. It's pretty cool to be part of this global in situ bake-off sale. That's pretty great. So I think what we've seen today with these talks is that depending on the spatial biology question that you want to ask, it vacillates between 2 metrics. So that's plex and resolution. And so at the moment, there's no silver bullet technology. Anyone who tells you different is trying to sell you something. So we think, though, here at Nanostring, we have 2 complementary solutions that can answer any spatial question that you have at any scale, right? So if you need whole transcriptome type of full unbiased kind of discovery, that relies on a profiling type of platform, which our GeoMx instrument can do. So we're honing into a region of interest on the tissue, a specific feature that you're interested in, and we can profile whole transcriptome information out of that, but it cannot go down to single cell resolution. If you require single cell resolution or subcellular resolution, that requires an in situ platform, which we've heard about several today. That is based on an imaging type of approach, and there are issues with spectral overlap and optical crowding. So at the moment, we cannot get up to whole transcriptome. And I know researchers are greedy. We'll get there someday. So the differences between the 2 platforms, I've already sort of alluded to those already. So on the Geomix side, we can do whole transcriptome. This is unbiased biomarker discovery. If you're looking for specific signatures within tumor and microenvironment, for example, it's a high throughput system. So this is your workhorse system, right? FFPE tissue, this is fully automated as well for sample prep with the LicaBond RX. You can process up to 12 slides per day, TMAs. You can get through a lot of samples all at once. Cellular resolution, it's cell enrichment or multicellular kind of approach, and we're looking at the differential expression between different samples. On the CosMx side, obviously down the resolution is a lot finer. You can do an entire slide scan, so a 300 millimeter squared area of tissue is what we can profile on our image on the CosMx instrument. It's a fully multi-omic platform as well, and we're looking at the differential expression or changes in interactions between individual cell populations. So for the Geomix instrument, why it's important, we can do feature-driven profiling. So if you've used sort of an array-based kind of approach, that gives a nice high-level overview of the tissue, but there are gaps in between the areas that you can profile. So with the Geomix instrument, what we can do is we can outline specific substructures in the tissue. So here we see some strata in the hippocampus of a mouse sagittal brain section, and we can do a full transcriptome pathway analysis out of that so you understand what that functional unit in the tissue is doing. And if you want to do a deep dive into single-cell subcellular, you'll move over to the CosMx. So brief introduction to the Geomix instrument. I know a lot of you are familiar with this. So a very simple in situ hybridization kind of workflow. You can place 4 samples on the Geomix instrument at once. We stain with morphology markers to outline the tissue architecture and cell populations of interest up to 3 colors. And then we also stain with proprietary reagents for protein and for RNA. So this is a non-poly-A kind of capture approach for the in situ hybridization up to whole transcriptome, and we can profile up to 147 proteins on the system. We select regions of interest on the tissue, which you can see in that sort of middle panel there. We shine UV light over that area, so it's a non-destructive kind of laser micro dissection of that tissue. And those barcodes are then collected and then processed for downstream on either an Encounter system or an NGS Illumina sequencer. So some just brief updates for the Geomix instrument. We have content for protein and for RNA, but we also have custom assays as well. So up to 400 genes, you can create custom content for any species. You can also spike that into any of our existing assays or run them as standalone. So the system, Geomix and CosMx, will sit downstream from a LicaBond RX, so your sample preparation time will be reduced, which will increase the efficiency of your workflow and also your throughput on the system. And we've also launched a new protocol that's called Spatial Proteogenomics, so we can detect both RNA and protein on the same sample. That's up to whole transcriptome, 147 proteins on a single slide. All right, so now I'll talk about the SMI instrument. This here is empirical proof that we have a marketing department. But the CosMx instrument, as you can see, it's very celestial in nature, so it's the CosMx. Internally, we call it the SMI. And in that vein here, this is pretty exciting. This is a 5-micron thick FFPE tissue. We're looking at 500,000 cells in this particular sample and over 1 billion transcripts. So what's exciting here is it's molecular mapping. We see these individual RNA species in individual cells located into the nucleus, cytoplasm, and the membrane. So just some metrics about what we can offer on the CosMx. We have a demonstrated 1,000-plex panel, and this is increasing. Our SVP, Joe Beecham, is in the lab right now increasing that, so pay attention for post-AGBT kind of updates. High sensitivity, our chemistry is exceptional in detecting low copy numbers. It's multi-omic in nature as well, so we can detect RNA and protein on this platform in both fresh frozen and FFPE tissues. And as I mentioned before, FFPE tissue is really our wheelhouse and where we sit. High resolution, we have subcellular resolution and three-dimensional analysis from 50 nanometers in the XY direction to 200 to 400 nanometers in the Z direction, excuse me. And all of this analysis will be enabled on the cloud. Geomix also as well in Q4 of this year. So in terms of the sample types that we can accommodate on the system, we've run almost 40 different projects, or 50 I think now, for customers, and so we're increasing the capacity. But we can run FFPE and fresh frozen on TMAs and skin tissue. Those top 2 ones are from projects in Australia, which I'm very happy about. And also we can run organoids and fresh frozen tissues from mouse brains as well. In terms of the chemistry, this is one of the differentiation factors between our platform. So on the left-hand side, we have a native mRNA species there, and we have a target binding domain, which is roughly 35 to 50 nucleotides. We tile 5 to 8 of those probes along an mRNA transcript. We only need 1 of those to bind. So what that means is that we can target highly degraded FFPE tissue in the mRNA species. In terms of the readout domain that you'll see there on the right, you see there's different blue colors. So we use a 4-color combination to identify each analyte. So we go through 16 rounds of cyclic hybridization and imaging. In 4 of those rounds, you'll have one of those reporters that will bind. So it's very similar to what we saw earlier in terms of barcoding. And then on the protein side, we have a series of antibodies, and then the readout domain is bound to that via a UV filtered cleavable linker. 1 minute, okay, gotta get through stuff. Go quickly. So the end of a single-cell RNA-seq experiment is your UMAP. What the CosMx can do is now it takes that X and Y direction where you can identify your novel cells and then recapitulate that into space. So you can see where they're actually sitting, what cells they're sitting next to, and how that changes their interaction. High level of how we do this, so we do that transcript localization through that cyclic hybridization, as I mentioned. We stain the tissue with morphology markers as well, and a nucleic acid stain, and then we use a cell segmentation algorithm to outline individual cell populations. So I'll just skip through here. This is a non-small cell lung carcinoma sample. This is available for you to look at, but what we can look at is cell type mapping, and we can also look at cellular micro-niches or cellular neighborhoods, which was also discussed today. We can take those cellular neighborhoods and put them into a UMAP so you can identify them. We can also do receptor-ligand interactions as well, and pairing, so we have over 100 different of those combinations. What you can see here is different patients on the x-axis there. On the y-axis are different combinations of ligands and receptors and variations between patients. We will have pre-validated content up to 1,000-plex and 100-plex for RNA species and up to 100-plex for protein. And there will be custom options available to complete custom de novo up to 1,000-plex in 2023. So you can download this publication. It's available on our website, and we also have a data set that's also available as well that you can play around with that through your bioinformatics pipelines. What's interesting to note about that data set is that we profiled over 800,000 cells, and we captured 96% of those passing QC. Median transcripts in these were 256, but we can max out at 2,000 transcripts per cell. What I want to note here is that the RNA quality of these, so we don't check RIN scoring or do DV200 on our platform. These were 3 down to undetectable for those. This is just a snippet of our 108-plex protein panel that was released at AACR a few weeks ago. This will be available on our website for you to have a look at as well. Just one more slide here, Paul. We got a fresh frozen 1,000-plex mouse neuroscience panel. This is fresh out of the R&D lab, and we did some NB clustering analysis for that. And what you can see here is that you can isolate and look at each of those individual cell populations as they're dispersed across the tissue. So thank you very much, and I'll pass it over to Sarah. Thank you.